In this section, we are going to be looking at tips that can help you get you exercises on genetics right. Genetics is a very attractive and popular chapter, but unfortunately, many students end up getting it wrong. We are going to guide you on how to understand questions. This presentation will not be very useful to a student who has not yet studied the chapter. It is worth noting that genetics, as we know, is based on Gregor Mendel's principles of heredity. As such, the program of genetics is broadly divided as Mendelian genetics, based on Mendel's laws of inheritance, and inheritances that deviate from Mendel's laws. Mendel's laws of inheritance include the first law of segregation, which says that each individual has two alleles for a gene, which separate during gamete formation, so that each gamete carries only one allele. The second is the law of independent assortment, which states that genes for different traits assort independently during gamete formation, as long as they are on different chromosomes. Third is the law of dominance, which says that in heterozygous individuals, one allele, the dominant allele, masks the expression of the other, the recessive allele. Mendel's principles set the basis for studying inheritance. However, the organism that Mendel studied before laying down these principles, that is the garden pea, tended to be more an exception rather than the rule. Thus, the inheritance of many characters, especially in animals, tend to deviate from Mendel's laws. Some of these include incomplete dominance, in which neither allele is completely dominant, and the heterozygote shows an intermediate phenotype. Codominance, in which both alleles are fully expressed in the heterozygote without blending. Linkage and genetic recombination, in which genes located close together on the same chromosome be inherited together, violating the law of independent assortment. Sex-linked inheritance, in which genes located on sex chromosomes, in X or Y, show different inheritance patterns between males and females. Epistasis, in which one gene masks or modifies the effect of another gene. Lethal inheritance, in which there is embryonic death of some genotypes leading to modified phenotypic ratios. Other deviations include polygenic inheritance and pleiotropy, multiple alleles, penetrance, and environmental effect. That is the epigenetic effect on gene expression. By having this overview, you can carefully check them out to know which one it is in the exercise you are treating. Now let us look at how you can understand your questions and bring out the necessary information. Let us quickly look at the notion of the gene and allele, which is crucial to getting your answers right. Assuming that you know the definitions, a clarification that can help you define alleles in a cross is that the gene goes used for the character, while allele determines the specific version of the character, for example, the gene for flower color and the allele for pink color or white color. The next thing to do is to identify the character and the type of inheritance. The character helps you to identify the alleles. This often seems easy, but for questions with the inheritance of recessive diseases like hemophilia, albinism, sickle cell, and color blindness, it becomes tricky. We cannot say that let a letter represent the dominant allele for hemophilia. That will make no sense. Rather, identify the character and you will find the dominant allele. For hemophilia, the character is blood clotting that fails to go through. Alleles can be for normal clotting and then for hemophilia. On the other hand, identifying the type of inheritance helps in two ways. First, to know how to represent your crosses, that is, the letters to use and how to write them. And secondly, to know how to interpret your genotypes to give phenotypes during the cross. To identify the type of inheritance, the question can give you directly. If not, look at the F1, F2, and the test cross phenotypic ratios. We will see these ratios later. Many questions, especially on linkage and gene interactions, often include test crosses. To identify that a question requires you to do a test cross, you will be told, for example, that two pure breeds were crossed and their offspring were crossed with an individual having recessive characters. Note that the questions often list characters, and you are the one to know that they are recessive. For example, tall and yellow seed pea plant were crossed with dwarf and white pea plants. The resulting plants were tall with yellow seeds. The F1 individuals were crossed with dwarf and white pea plants. This directly tells you that a test cross was performed. You may also need to form gametes from a genotype you should already know how many types of gametes will be produced. Count the number of heterozygote loci are there in the genotype. Each heterozygous locus contributes 2 gametes. For example, the genotype A, B, B, C, C tribrid cross will produce 4 gametes. The locus for capital A and small a can produce gametes with allele capital A or small a, same for B. This helps you to know the ratios of each gamete, which you will use to calculate the ratios of the genotypes and then phenotypes. Determining the parental genotypes is crucial for getting your answers right. To get the genotype, look for keywords in the question statement, including the following: pure breeding, pure line, etc. These mean that the individual is homozygous. The ratios of the F1 will tell you which phenotype and thus the allele dominant, recessive, codominant, etc. The word hybrid implies heterozygote. Also, F1 individuals obtained from pure breeds are heterozygous. The words mutant in wild type often imply homozygous recessive. In inheritance with dominance, a dominant phenotype has 2 possible genotypes: homozygous, dominant, and heterozygous. A recessive phenotype has only 1 genotype, homozygous recessive. Thus, from the question, it is often easier to know the genotype of an individual showing a recessive character, that is, homozygous recessive. We have come to the end of the first part of this presentation on tips on genetics. In the next presentation, we are going to continue with the second part on how to identify the ratios and use them to understand the type of inheritance to use. Like, follow, and share so as to get notifications of new presentations.